Great, thanks for that introduction. I have actually never attended an MIA seminar before. I've watched several of them online. But just to get a sense of who is here, could I get a show of hands? Who knows what an enhancer is? Okay, that's great. That's a great start. How many people do some experimental biology?
Like
wet experimental biology, yes. Okay, great, excellent. My background, I studied computer science in undergrad, but in Eric's lab here, I've been mostly focusing on molecular biology and experimental genomics because in my undergraduate studies, I became very interested in how gene expression is regulated and how it works, and decided that I really need to be able to do these experiments in order to pick apart these processes. So the talk today is really a little less about models, inference, and algorithms, and more about molecules, interactions, and aspirations. And the goal here is for this to be an interactive discussion, and my goal here is going to be to share a little bit about my worldview about how we can tackle some really big problems in molecular biology and genetics, and sort of interface between experimental and computational approaches to pick apart these processes. I think the sort of, I'm going to give one example of a problem that we've been working on lately in the Lanner Lab that sort of goes through this, I guess, paradigm of trying to understand a molecular process in a very systematic and systems level way, figuring out which data we need to actually model or understand this problem de novo, and then applying the appropriate modeling techniques in order to understand it at a systems level. So I think there are a number of other problems besides the one I'm going to talk about today that can be approached using the same general approach, and I'd be excited to continue conversations with anyone here about potential collaborations to tackle some of these other problems using a combination of experimental and computational approaches. So the title of the talk is Mapping the Regulatory Wiring of the Genome. And before I get into exactly what I mean by that, I'd like to start with the motivation for why we'd like to understand molecular processes of the cell. This is just one particular motivation that stems from, I guess, the last few decades of medical genetics, of which the goal has been to understand the molecular basis of disease. Starting with the sequencing of the Human Genome and the Human Genome Project, which of course Eric was very involved with at its inception 30 years ago. The idea was that by understanding the genes that are mutated in people with diseases, we can understand and narrow in on the molecular basis for disease. And this has been particularly challenging in the case of common diseases, where it's not one particular mutation or one particular genetic variant that ends up causing the disease, as in the case of many Mendelian diseases, but it's a confluence of many different variants of small effects that together shift your overall susceptibility to disease. The picture in about 2000 was that we really didn't have any good handle on understanding the genetic basis of common disease. There was a lot of potential insights in our genomes, and that could be gleaned if you were able to compare the genomes of many people with disease and many healthy people. But that would involve determining sort of which loci in the genome might contain causal variants, understanding which variants actually tweak the function or expression of a gene, understanding the gene, its role in cellular pathways, as well as at the organismal level, and then ultimately leading to a therapeutic hypothesis about some particular molecule or gene that we could drug in order to modify disease risk. So a huge amount of effort in the last 16 years has gone into, of course, finishing the sequence of the human genome. That was a major barrier. Mapping genetic variation in the population. So this is the concept behind HapMap, is to identify sort of the haplotype structure among different individuals in the population, and ultimately to do genome-wide association studies where you say compare the set of variants in thousands of people with disease with thousands of healthy people to identify regions in the genome that are associated and actually contain some causal genetic influence into disease risk. So it turns out that at this point in time, we have thousands of such, I guess, insights that have made it to this stage, where we know that the locus in the genome that harbors some genetic variant that influences disease risk. But there are very few where we've actually made it down to through this full pipeline of understanding and converting that into a therapeutic hypothesis, by which we need to understand the variant that's involved, the gene that's being regulated, and the pathway that that gene acts in. And sort of the major goal in human genetics now is essentially to move these thousands of loci that are associated with various or dozens of different diseases to this spot here, where we can actually hand it off to a pharmaceutical company to go develop a drug. So what are sort of the remaining steps that we need to solve, which are every bit as challenging as sort of these three here at the top of sequencing the human genome in the first place? So the challenge now is that we need to understand the functions of genes, and we need to understand the functions also of the non-coding genome, which regulates genes. This is what I'm referring to when I say the regulatory wiring of the genome. We need to understand the genetic code that not only encodes gene function, but also encodes gene expression and the interactions between genes, pathways, and cells. So where are we in that process? This is coming from the perspective of, again, from human genetics, we have thousands of loci, each of which contains several variants, any one of which could be causal in terms of influencing disease risk, and we need to understand essentially what each one of those variants does. So if you're so lucky as to have those variants end up in the coding regions of the genome, we actually can do a pretty good job of understanding the functions of variants there. Since the 1960s, we've understood the triplet genetic code, which converts the language of nucleic acids into the language of amino acids. And so we actually can recognize variants that affect the DNA sequence and understand and predict their functions on protein. It turns out that most of the genetic variation associated with common diseases actually does not fall in the coding regions of the genome, and most of it actually lands in the non-coding region of the genome. And so I'm going to just put up a scorecard of how we're doing in terms of understanding those variants, the majority of variants that land in the non-coding regions of the genome. So the non-coding genome is 98% of our genome, and we actually don't understand the genetic code of the non-coding genome. Somehow the protein coding part of it got the name the genetic code, when really there's a vast amount of information in the non-coding genome that we don't know how to understand or interpret. So pieces of the non-coding genome might include promoters, which sort of assemble polymerase and cause it to fire to produce gene expression. And we can sort of recognize in the genome, we definitely know which gene is affected if you were to delete a promoter, because it's directly associated with the gene. We can't really predict or interpret the functions of variants that occur in promoter regions. There are a number of other different types of non-coding DNA regulatory elements. I'm going to focus mostly on distal enhancers in this talk, which we can sort of recognize in the genome. I'll get more into that in a minute. We don't really know how to predict which gene might be affected if you were to break an enhancer, and we don't really know how to predict the effects of variants in enhancers. There are other potential types of DNA regulatory elements. There's also an entirely other category of non-coding sequences that we might care about and that might play a role in disease risk, which is non-coding RNA regulatory elements, which would be variants that occur in the DNA but then are transcribed and affect the function of the RNA itself. Those might involve splice sites, splicing regulatory elements, transcriptional termination signals, which we don't know how to recognize, potential translational regulatory signals like microRNA sites or other types of signals in UTRs. And the language of all of these types of elements is pretty obtuse at this moment. We have some ideas about the molecular mechanisms that might contribute to these various processes, but we don't really have in any systematic way a way to predict the effects of any given variant. So I'm going to, like I said, I'm going to dive in a little more detail into the enhancer category and say, can we check a couple more of these boxes? And can we actually begin to predict which things in the genome are actually serve as enhancers, which genes do they regulate, and can we understand the effects of variants in those enhancers, and thereby perhaps interpret non-coding genetic variation that happens to land in these types of elements. Okay, so enhancers play an important role in regulating gene expression. And gene expression, of course, fundamentally is at the heart of the differences in behavior and functions of different cell types. So we have approximately 20,000 genes in our genome. We have 34 trillion cells in our bodies, many different cell types. Each one of those cell types expresses different combinations of genes at different sort of quantitative levels, and it's those combinations that really confer specific functions to specific cell types. So the way that these intricate expression patterns are regulated is thought to be through the functions of non-coding sequences called enhancers, as well as many other types of regulatory elements. But enhancers in particular are thought to be located distal to genes, but the sort of network connecting enhancers with the genes they regulate is thought to be complex. One enhancer can regulate multiple genes. One gene can be regulated by multiple enhancers. These interactions can occur over long distances, sometimes, say, over up to 2 megabases of sequence, meaning that it's difficult to predict which gene might be regulated by any given enhancer. And further complicating the matter is that different sets of enhancers are thought to be relevant to the same gene in different cell types. So for example, this blue gene here would be regulated by these couple of enhancers in cell type A, but a different set of enhancers in cell type B and a different enhancer in cell type C. You can imagine that this, from the perspective of interpreting genetic variation, this means that we not only have to understand the effects of a genetic variant on an enhancer, but we need to do that in a cell type specific manner, because it appears that there's a different code in each cell type that would affect whether this enhancer is used in the first place and also potentially the function of a variant. So this is a really massive network and matrix that we essentially have to fill in. There are 20,000 genes. They're thought to be maybe millions of enhancers. It's kind of unknown. Thousands at least of cell types, and the goal would be, in order to interpret the function of a genetic variant, we essentially have to understand this entire matrix. So, where do we start? So, let's talk a little bit about what an enhancer is and how it works. That's pretty critical to understand how we might pick apart this matrix. So, an enhancer, like I said, is a distal element. It's thought to encode a specific sequence that might include various short sequences called motifs that would be recognized by specific transcription factors, and that a set of transcription factors would assemble at the enhancer, and perhaps there's another set of transcription factors that assembles at the promoter. And then it's thought that these things physically loop or contact one another, and that's where the magic happens, where the set of transcription factors at the enhancer interacts with the set of transcription factors at the promoter and helps polymerase fire and drive gene expression.
Just to be clear, for the mixed audience, the transcription factors are themselves proteins.
Correct.
That are coming from other genes.
Yes, thank you.
Are there any cases where the enhancer affects other chromosomes on both sides?
There are a few weird cases that have been reported where that appears to be true. One of the interesting ones is in olfactory receptors, where you want to express one of hundreds of different olfactory receptor genes, which are located in clusters on different chromosomes. Turns out in olfactory neurons, those clusters on different chromosomes hang out in the middle of the nucleus together and coordinate so they only turn on one of those genes. I think that's the exception rather than the rule.
That's fine.
Yeah. Yes?
This model where the enhancer ends up being physically adjacent to transcription, something you observe, or something that you're, or this is the only plausible way that...
Sorry, could you repeat that a little louder?
Is this, how much of this is experimental observation versus what we think is a plausible way this could work?
Yes, that's a great question. So That is a very, so, okay.
What was the question?
The question is, how much of this is experimentally observed versus a model that for how this could plausibly work? Transcription factors and their role in driving transcription and their functions and enhancers have been studied for sort of 30 years at a like single transcription factor, single gene, single enhancer type level, where mostly from a biochemical point of view, where people have measured the interactions between transcription factors and DNA. There's sort of an overwhelming body of evidence would suggest that there's a set of proteins, transcription factors, that drive transcription. We know that these transcription factors sit at enhancers. One of the sort of enabling technologies recently has been chip sequencing, where you can actually essentially measure the occupancy of transcription factors in the genome and determine where they sit. Experiments, over the last 30 years on sort of a genetic level have been able to mutate some of these specific sequences that are known to interact with transcription factors. And you can know that if you mutate this particular sequence, the transcription factor no longer binds there and you no longer get transcription. So a lot of this, I think this particular picture here is very well supported by the evidence. I think the challenging part is that we don't have enough observations of this type of thing actually happening in the genome to really understand or infer any general rules about that might let us predict which enhancers regulate which genes. So for example, a few of the things that remain unclear are how exactly this looping occurs, or to what extent it influences the effect of an enhancer on a promoter. Another thing that's unclear is whether there are specific combinations of transcription factors that would sit at enhancers and allow them to talk to specific genes. That's unknown. And so those are kind of critical things to predicting which enhancers regulate which genes. But overall, this model is pretty well accepted and pretty well supported.
This is basically a verified hypothesis that it is through genuine 3D proximal interaction.
Yeah, so that looping part of it, I think, is just sort of emerging now. There's pretty good evidence for that in the last, say, couple of years, but I think this is generally well accepted. There are proposals that there are types of elements that act through other mechanisms other than looping, and I'm just going to say those are in a different category of elements, and I'm not going to talk about them today. Okay, so so there are a couple things that have happened in the last few years that make it possible to make it at least conceivable that we could solve this in a across all cell types and all genes. So one is the emergence of methods to map in a genome-wide manner this sort of chromatin state, which is to say there are assays like DNA hypersensitivity sequencing, which essentially measure how accessible any particular region of the genome is. We can think of this as a measure of how many transcription factors are sitting at any particular location in the genome. There are methods like chromatin immunoprecipitation sequencing, where you use an antibody against a particular protein or a particular histone modification and map the occupancy of that particular protein or histone modification across the genome. So, for example, that could reveal where all of these transcription factors are sitting, or where any particular transcription factors is sitting. And then there are other methods like Hi-C, which measure the three-dimensional proximity contacts of the genome. So for instance, this method can measure how frequently any 2 points in the genome are physically in contact with one another in a cell population. And so assuming that this model is actually correct and that it contains all of the relevant information that we might need, we actually have ways to measure many of these things on a genome-wide scale across many cell types. And so, for example, here's just an example of what you might see if you were to use one of these techniques, in particular K27-cell chromatin immunoprecipitation sequencing, which is this is a mark of active regulatory elements, including enhancers and promoters. And this is a 2 megabase region containing a few genes. The point here is simply that to show that across 8 different cell types and tissues, these marks actually change a lot, consistent with this idea that there might be different types of elements that are regulating genes in different cell types. And there are specific focal sites across this 2 megabase region that are particularly strongly marked by these chemical modifications that are indicative of enhancers or promoters. The challenge, of course, is that we don't know for any one of these given regions which genes they regulate. And the goal would be to be able to draw the network that connects each of these sites with the genes that it controls. So the major limitation to doing that as of a year ago was that it was really difficult to actually do the experiment that you want to test those connections. The experiment that you have to be able to do is to inhibit the function of the enhancer and read out the effect on gene expression. It's like very conceptually simple, but it was really difficult to do because it was difficult to modify the genome. So with the advent of CRISPR, of course, this became somewhat easier, but still rather challenging. And so the first step was to be able to develop an experimental method that would be able to functionally test enhancer-promoter connections and essentially to delete or inhibit many hundreds of enhancers and read out the effects on gene expression. Yeah?
Jesse, so it sounds like you're transitioning that scorecard from part where you say, can we identify enhancers, to can we figure out which genes are regulated by those enhancers. So before we leave that part of the scorecard where you're saying, can we identify enhancers, are we content with the idea that in order to identify enhancers, we effectively have to do an experiment? We can't just read the code as we could, gene discovery, for example. I guess you could do transcription and so on, but there are ways that you could just look at the sequence and infer whether there's a gene there or not.
Right. So
how close are we to that with enhancers, or is that ever going to be possible from your point of view?
Yeah. There are a few questions here. First, we're not moving past this point in the talk yet because without the ability to functionally test them, as in delete them and see what happens in gene expression, we don't know that they're an enhancer or not. So we have to be able to do that experiment both to connect them to the genes, but also to figure out if they're actually functional. So I would say that these marks tell us the possibilities for what might be enhancers. As to whether we can read the sequence and identify these things, I think that someday we'll be able to do that. But again, we need to do a lot of experiments first to figure out which things are actually functional. Then we can build models to actually learn those rules.
So once we have labels on specific regions that say yes, enhancer, not enhancer, in
Right.
this specific cell type.
Yeah, right. Then we should be able to build sequence-based models that would actually identify these. Again, the challenging part there would be to actually make that generalized across cell types because there's a different code in every different cell type. That would make it very challenging to potentially learn general models that could generalize to an arbitrary cell type. Okay, so here's the overview of what we'll attempt to do. We're going to attempt to experimentally map many enhancer gene connections so that we can figure out, one, what things are actually enhancers in the genome and which genes they regulate. Two, we're going to then attempt to compare that data with all of the other chromatin state maps or other data that exists and see if we can actually learn the principles of how enhancer gene connections are specified. And then ideally, if we understand those principles well enough, we're going to be able to build predictive models that could predict these in, say, new cell types or at new genes that we haven't measured yet. Okay, so starting with number one, I'm not going to go into a great deal about this experiment. It was actually recently published. This is the work of another graduate student in the lab, Charlie and I. And the idea here is to leverage CRISPR to be able to do the experiment that I mentioned a minute ago, which is to map and perturb thousands of different enhancers at once and measure their effects on gene expression. And so the particular format that this experiment takes is that we're going to pick a single gene and we're going to tile guide RNAs around, say, every possible site around this gene over some, expanse of the genome. And we can actually do, say, hundreds of thousands of different guide RNAs in the same experiment, in the same pot. And the way that we do that is we take a population of cells, we infect these cells such that each one expresses a single guide RNA, and then we perform a phenotypic selection, for instance, sorting cells based on the expression of this gene. And then we sequence the distribution of guide RNAs before and after that selection. So, for example, in this case, I think the dark purple guides have dropped out of this population, which means that, for instance, they might be affecting a regulatory element that regulates the expression of this gene. So essentially what this gives us is a way to map thousands at once. This is a prerequisite for understanding these types of connections at scale, and it is actually quite remarkable given that, for instance, in 2 years of my PhD, I spent painstakingly knocking out 20 elements and measuring their effects on gene expression. And this means that in a month, you can do thousands. So this has been really a game changer for us, and we've set about applying this method across dozens of genes. I'll just show you an example of one gene. This is the same locus that I showed a moment ago, centered on this important transcription factor called MYC. We tiled guide RNAs across a large portion of this 3 megabase region. So there are hundreds of thousands of individual guide RNAs in this region. And what I'm going to show you here is the CRISPR score. Essentially, higher on this axis means that you have a stronger reduction in the expression of MYC when you inhibit it. In other words, high on this axis means that you're a strong activator of MYC. And it turns out, if you look across this 3 megabase region, there are 7 specific sites that actually affect MYC expression, which are labeled here. Well, actually 8. One is the promoter itself, so that we kind of expected. But the surprising thing is that there were 7. We didn't know what number we might expect. It could have been hundreds, it could have been 1, it could have been 0. Turns out there are 7. And they are, in fact, spread across a very large region. So these are up to 2 megabases away from the gene, which is quite surprising and unusual. And if we then go in and inhibit each of these elements individually and measure the effects on gene expression, they have sort of a variety of quantitative effects. So for instance, inhibiting this guy out here leads to a 10% reduction in MYC expression. Inhibiting this guy here leads to a 50% reduction. Yep.
So if you look at the E5 score,
Yes.
It's actually about the same with something that you find in the middle,
Yep.
which is not.
Yeah. So here I'm showing essentially the mean of 20 guide windows. What I'm not showing is the significance, and it turns out that this is, high mean but large variance, so it's not significant. So these are the significant ones. Yes. Good question. Yes.
How big is your window for this tiling? Like when do you?
We don't really know. We don't really know. We chose to hit these regions because in the slide that I showed you previously where we had mapped K27 acetylation, these were the regions that had K27 acetylation. But you could imagine that maybe we should extend the window farther, up to 10 megabases or something. And we didn't. There had never been any enhancers that had been reported to be, say, 10 megabases away from the gene, and so we didn't think it was reasonable to do that. There are other reasons why we picked this particular window related to sort of the structure of the 3D genome in this region, where this whole segment here is part of what's called a topological domain. It seemed reasonable to look within that region. As we've done more of these experiments, these very long distance interactions over 2 megabases are very rare. So we don't think we're probably missing many by choosing this particular window, but you're absolutely right. I mean, we don't really know. Yes?
Do you think it would be worth it at all to fill in the regions that didn't score with the K27 acetylation?
So these regions cover many regions that don't score, or that are not K27 acetylated, and all of the regions that do score are K27 acetylated. And we have not found so far any regions that score that are not K27 acetylated. So, so maybe, but there are different ways that you can imagine doing this particular experiment. We used a epigenetic repressor called CRAB. It might be better to go back and do this by deletion. There are various experimental variations that we're working on, and to try to understand whether there are things that we're missing in this assay. So, so we've done this at this point on several dozen genes. And by combining this data with other existing data that we've generated previously, we have now a collection of approximately 500 putative enhancer gene pairs, where a putative enhancer is going to be a DNase hypersensitive site, 50 of which actually affect the expression of the gene, and about 450 of which do not affect the expression of the gene. And we're going to use this data set to try to learn how the genome encodes specificity for these connections. So let's compare this results of, in this particular locus, with the chromatin state maps that I mentioned before. So you look at that, here's DNase hypersensitivity, here's K27 acetylation. As Aran asked a moment ago, how did these line up? It turns out that the strongest peaks here almost line up perfectly with the strongest sites of K27 DNase hypersensitivity. So this was encouraging because it meant that that picture that I showed at the beginning seems to be correct, at least in part, where the things that have transcription factors binding and have marks of transcriptional activity are in fact the sites that actually affect the expression of genes. So that's good. What was interesting is that, and kind of not known, or it was a little unclear before this, was that it should correspond necessarily to the sites that have the strongest quantitative values of these measurements. It wasn't really known how to interpret the quantitative values in these experiments, and in fact, consortia like ENCODE had gone and, say, defined peaks in this data. And if you do that, you see there's essentially about 100 peaks of DNase hypersensitivity in this region, which were essentially all annotated as putative enhancers. Turns out that the ones that matter are the strongest ones. We'll get back to what that cutoff is in a minute, but so that was an important insight. And so I'm going to start drawing this locus like this, where the size of this circle indicates, I guess, the strength of these signals here. Yes?
Remind me what your cutoff was for those 7. Was it just sort of statistical significance in your...
Yeah, right. So there could be, it could be
a tail and a continuum.
Yes, it could be continuum where these more minor peaks have very small undetectable effects. The smallest effect that we detected in our assay was a 10% effect on gene expression. So, but it's possible
that's
that
limit. That's your measurement limit.
That's a measurement limit. So it could be that there are others that regulate at much smaller quantitative effects. Yeah.
How do you think about the height of the EHS peaks? Like, is that for you? Is it influenced by the number of cells in the population that have that region open? Is it the openness average? I mean, I'm not that familiar with the experimental procedure, so how do you think about the height of the peaks?
Right, so it comes down to sort of the details of the assay, and at some extent it's guesswork. I think the things that you mentioned were essentially how I think about it. The DNase assay is measuring, is using a nuclease to go in and cleave DNA, and it can only do that when the nucleosome is sort of removed and there's transcription factors sitting there. So essentially, when you are cutting, it's a binary assay whether you cut or not. And so what you're measuring is, I guess, the frequency of cells in your population at which you can cut. And that would be a function both of how often that region is accessible across cells. Okay, so I'm going to draw this locus like this. Size of the dot is approximately corresponding to the quantitative signal in these regions, and these lines are going to connect them to the genes that they actually regulate. So the quantitative signals in these two assays is important, but it's not really sufficient for the reason related to this question, which is that we don't really know which window or which distance we should actually care about. Do these enhancers then regulate these genes as well, or how would you end up connecting these particular enhancers to regulate this specific gene as opposed to some of these other genes? Here we want to pull in the 3D conformation of the genome. And this is a depiction of what is measured by Hi-C, which is essentially a 2D heat map specifying the frequency of contacts between any two regions. So if you were to imagine looking at the heat map down this line, this is essentially the contact frequency of each of these sites along this region with the MYC promoter itself. So one of the hypotheses in the field has been that you should care about these what are called topological domains, which, as you can see, are sort of these regions where there's increased frequency within the domain relative to, say, between domains. As it turned out, that this part, the presence of the domain, didn't actually seem to be predictive, although it did a good job of determining which enhancers would actually regulate MYC. It turns out only a subset of these enhancers regulate each of these two genes, and so this wasn't a particularly good model. So instead, we thought we sort of thought about this model that I presented at the beginning, where enhancers are floating around in space. When they contact each other, that's when the magic happens. Perhaps each enhancer has a different strength related to how frequently or how often the transcription factors are actually bound. And so if you were to imagine putting those together, you could create a simple model where each enhancer would be described by its a quantity that I'll call its activity, which is how much transcription factor stuff is there, how many RNAs would be produced when it bumps into the promoter. And then a second parameter would be its probability of contacting the promoter relative to other regions. So, for example, this large dot here might have a large activity but small proximity because it's farther from the gene, whereas this other smaller dot would have a smaller activity but perhaps a much larger proximity, which could give it a larger overall impact on expression. And we could imagine that in this simple model, you might then be able to predict the quantitative effects of a particular enhancer on gene expression if you know all of the other enhancers that this particular gene is listening to. Yeah.
Do you always see those lines for the topological domains? I mean, I remember the off-diagonal spots, and then, but you see, like that dark.
This line?
Yeah, that like this site is touching all the sites.
No, you don't always see that line.
Kind of special.
That's a slightly unusual feature. We don't really know why that line exists, but it probably has something to do with CTCF being there. Okay, so right. So the important part here is that we're going to actually just measure the intensity of the Hi-C signal in this matrix. We're not going to say look for the presence of focal loops or lines. We're going to just ignore the sort of features, the structure of the data, and we're just going to pull out the bin corresponding to the contact frequency between these sites. Sure. And then for activity, we're going to pull DNase hypersensitivity and P27 settle and create a number that represents the activity of each enhancer. And we're going to see how well this model does at predicting which enhancers regulate which genes, and sort of starting with a very simple model for how this might work. So, so here now I'm showing, again, the same locus with 3 different genes now. MYC is here. What I'm showing here is, imagine taking a column or line from that 2D matrix and plotting it on the y-axis. So for instance, this red line represents the proximity of each of these regions to MYC, whereas the yellow line represents the proximity of each of these regions to PBT1. For each of the 7 enhancers, we can calculate a value for activity. For MYC, we'll essentially take each of these regions, read out the height of this red line, and say that's the contact probability or proximity. We'll multiply those together to get a predicted effect on the gene, and compare that to the measured effect, which actually corresponds quite well. And I'm going to sort of tally that up over here, where the predicted effect is on the x-axis and the measured effect is on the y-axis. Okay, so here's one gene. Now we're going to look at another gene in the region. PBT1 has sort of the same activity values because the enhancers are the same. We're going to look at the same set of 7 enhancers. We're going to pull out the proximity values. In this case, E3 and E4 are sort of in this key critical window here where the Hi-C contact probability really spikes. And so they have much higher proximity values than the other enhancers. And that leads to much stronger predicted effects on the expression of this gene. And it turns out that when you actually measure the effects of those enhancers on the gene, that corresponds really well. So one more gene out on the end. Again, the E5 here happens to land in this really strong region, leads to a strong predicted effect, which corresponds with the measured effect. And so that is actually starting to look pretty nice. Yeah?
Have you ever looked at the linear distance instead of IC distance? Because IC distance is highly correlated with the linear distance, and do you see better or worse?
Yeah, so the strongest signal that you see in high-C data is sort of related to the exponential, is essentially an exponential or power function of linear distance. If you plug that in instead of proximity, you still do pretty well because, you know, most of the signal is coming from this peak. But you do miss things like out here where these enhancers actually show sort of a focal enrichment of interactions with MIC. And so you would actually miss these enhancers if you weren't to use the actual high-C data. So, yes?
Seems like the proximity is one of the strongest effects here. I'm just curious if your model is more predictive than just predicting based on proximity alone.
Yes, it is. I focused on these 7 enhancers here because we had found them to regulate MIC, but remember that there are hundreds of other ones, and we wouldn't have been able to distinguish these 7 from the other 100 if we didn't have the activity measurement. But in terms of their relative effects, you know, they are also playing a role in creating this nice correlation. If you take this away, this line looks worse. Yes?
E3, E4, and E5, they are actually located like on promoters of PBT1 and CCDC26. So is it possible that sometimes genes may hijack other promoters as their enhancers?
Yes, so it turns out that many promoters can also act as enhancers for other genes. In this particular case, E5, for instance, is about 2 kb upstream of the transcription start site of this gene. So it would not necessarily be called the promoter of that gene. But in the case of PBT1, I think E4 is essentially right on top of the transcription, one of the transcription start sites of PBT1. So you might call it, in fact, a promoter. This is an important complexity that still remains to be worked out. Some promoters appear to act as enhancers, and some promoters don't have the capability to act as enhancers. And our model actually doesn't do a great job of distinguishing between those possibilities at the moment. Okay, so so we can do actually a pretty good job of predicting the quantitative effects. In fact, this let's see here. In fact, it's kind of surprising that this really simple model can actually predict some of the weird behavior that we observe in this locus. So, so for example, here's the sort of full connectivity diagram of this region based on the experimental data that I just showed. All 7 of these enhancers regulate MIC. 2 or 3 of them rather regulate PBT1, and only 1 of them regulates this gene here. So effectively, what's happening here is that these enhancers, this enhancer here is appearing to skip over CCD26 and PBT1 to regulate MIC. And this is a phenomenon that has been observed in other loci and has been sort of used as evidence that there must be some biochemical compatibility between specific enhancers and promoters in order for this enhancer here to skip over several genes to regulate a more distal one. But what this model, this simple model suggests is that simply the location of different enhancers and genes can create differential sensitivity. So for example, this enhancer has the capability to be compatible with this promoter, but this gene has this very proximal enhancer that's essentially contributing almost virtually all of its input signal. And so this enhancer here might have a small undetectable effect on this gene, but has a detectable effect on MIC. And so the specificity and these phenomena of skipping is created by not by specific transcription factors interacting with each other, but rather just simply due to the sort of emergent effects of the proximity contacts of where you would put these various enhancers relative to different genes. Yes?
I realize it's not such a great question, but why do you think you might have like enhancers laid out like that for MYC? Why would that be a good strategy?
That's a great question. There are a few different reasons. One reason could be that MYC is used in many different contexts. We have 34 million cells in our body. Not all of them are proliferating, but essentially every one that's dividing has to express MYC, and perhaps at a very specific level. So you essentially need an enhancer for say 1 enhancer or 7 enhancers for every cell type in our body, which means that you need a fair amount of genomic space to encode that information. So MYC is one of these unusual genes that lives in the region where so both of these are actually link RNAs. Neither of these are protein coding genes, and neither of them appear to have functions as RNAs. They're just, there might be something related to their transcription, but essentially this entire 2 megabase region of the genome is dedicated to regulating MYC. And actually there's a megabase over here that's also dedicated to regulating MYC, but just isn't active in this particular cell type. So 3 megabases of the genome is dedicated to regulating this one really important gene. And if you look across different cell types, their enhancers spread out all across this entire region in different cell types. So it might just be a space constraint thing. It could also be that you want to put enhancers farther away from MYC because it creates temporal variance. So for instance, this guy floating out 2 megabases from MYC only hits MYC very rarely, let's say every 10 minutes or something. So that essentially is, even though the mean might be the same as if you put a weaker enhancer over here, the variance is different. It's possible that that's important. Okay, so what this means essentially, as I showed you in this one locus, is that we have a model where we can take as inputs genome-wide chromatin state maps, which include DNase hypersensitivity, K27 settle, and Hi-C. We can stick them into this actually very simple model that essentially multiplies a few numbers, and that has the potential to output what we set out to develop at the beginning, which is a regulatory map that describes the connectivity between enhancers and genes. If we apply this model to all of our data and create a predictor that's essentially calculates the predicted effect and then draw a cutoff at some particular region and say, enhancer gene pairs on one side of the cutoff are going to be predicted as actual regulatory interactions, and below the cutoff will be predicted as not regulatory interactions. If you simply use linear distance alone, you would get this. If you use closest gene alone, you would get this in a precision recall curve. And if you use this activity times contact probability model, you can actually do a pretty good job, say about 85% positive predictive value at a recall of 85%. So there's a lot still to explain, but this actually does remarkably well given the simplicity of its formulation. So there of course is much to be explained here, but I think we're making good progress to understanding it. There are a lot of additional factors that we need to understand that might improve the performance of this model further. It's possible that there is a contribution of biochemical compatibility that might tweak the effects of specific enhancers on specific genes. We think that that the presence of these particular features in the chromosome confirmation data, like TADs and loops, are simply going to tweak the proximity values between various enhancers and genes, and that you really have to understand all of the enhancer inputs to a single gene to be able to predict whether any given enhancer regulates a given gene. And so this means that the organization of enhancers and genes in the genome is of incredible importance, and we're still working on understanding what that actually means in terms of why the genome would encode it in these different ways. So in the last few minutes, I'd like to show an example of using and applying this model to interpret non-coding genetic variation associated with disease. So I've showed you an example in a particular cell type, and we'll go back and look at this locus one more time. Again, looking at the MYC locus, we had mapped initially the enhancers K562 cells, which is an erythroid cell line. And again, here across 7 other cell lines, the enhancer landscape varies dramatically. And again, this is across 3 megabase region. And it turns out if you do genome-wide association studies, there are all sorts of regions that pop up as associated. And it wasn't exactly clear that all of these should necessarily regulate MYC, but now, given this model that, enhancers even out to 2 megabases away can actually regulate MYC, it seems that many of these regions might actually be associated with various, say, cancer or immune phenotypes because they're affecting cell type specific enhancers that regulate MYC. We can take a couple examples. For example, this one here is a strong enhancer in a B-cell lymphoma line. If you look, there's a GWAS association with Hodgkin's lymphoma that contains a series of linked SNPs that overlap precisely this DNase hypersensitive site, as well as several conserved nucleotides. So presumably one of these nucleotides is actually contributing to susceptibility for Hodgkin's lymphoma by affecting the function of this enhancer and thereby affecting the function of MYC. And in fact, our model, if you plug in the data from this particular cell type, does predict that this enhancer would regulate MYC expression. Similarly, there's a variant out here in this region close to these enhancers that we found in K562 cells that is engaged in this long-range loop with MYC, where a single variant overlaps this very strong enhancer in chondrocytes, and this variant is linked with height. So we could predict that this variant is actually influencing height by affecting the proliferation of chondrocytes or a chondrocyte-related cell type. We're thinking about ways that we could apply these predictive models to, for instance, do rare variant burden tests. If we have, if we can associate regions of the genome with particular genes, then we can start to understand and look de novo for variants that might influence a particular phenotype or rare regulatory variants. So just to return to the scorecard in the last few minutes, we're doing a better job now of figuring out which things in the genome are actually enhancers and which genes are actually affected by those elements. We still don't really know how to predict the effects of particular variants, at least not based on what I told you today, but through other technologies, particularly synthetic DNA technologies, where we can synthesize different sequences and test their enhancer activity in massive parallel assays, we at least have the capability to generate the data that we could use a similar approach where we, for instance, could think about how transcription factors might interact with each other on a molecular level and start to build computational models based on high-throughput genetic data to start to put together and be able to predict the effects of variants. Of course, there are many other pieces of this puzzle that remain unexplored, but essentially we have all of the experimental technologies that we would need to generate the data that would allow you to build good models of these processes. And it's simply a question of digging into each of these each of these pieces, which each actually has its own specific sequence code and specific molecular set of rules that might allow you to predict where they are and what genes they affect. And we can start, essentially filling in each one of these boxes through a similar set of processes. So I think we're actually the challenge of mapping the full regulatory wiring of the genome, which requires understanding the functions of non-coding variants and understanding the function of those variants in a cell type specific manner. I think that's actually within reach in the next few years. And I think that all of the technology on the experimental end exists, all the technology on the computational end exists, and it's all about making those connections to fill in these boxes and to understand put together the right pieces in the right combinations. So I'm incredibly excited about the next few years and what this community might bring to that effort. And I'd be excited to continue this discussion now or at a later date. So with that, I'd like to thank the team that made this possible, particularly my mentor, Eric Lander, as well as a graduate student in the lab, Charlie Fulco, and a computational scientist in the lab, Ray Jones, who did a lot of the heavy lifting on the analysis and experimental work that I described today, as well as the rest of the Lander lab and collaborators in various disease areas. Thank you very much for your attention, and I'll take any questions now.